Oh, thanks. Yeah, sorry I was running a little bit late. I'm going to run this. This is a slideshow. I'm going to run it in notebook mode because I was having a little problems with it. Yeah, so this talk is about the BioJulia organization and some of the work we've been doing and some of the work we're going to do. So why, in particular, am I interested in Julia? There's a lot of reasons why you might want to do biology in Julia, but for me, it basically comes down to this. So basically, around about 2007, the cost of doing DNA sequencing started plummeting. The technology is more or less the same as what it was in 2001 when we first sequenced the human genome, but we figured out how to do it massively parallel, essentially. So once these started coming on the market, the price of sequencing a genome in a decade went from $100 million to sequence the first human genome to now a few $1,000s these days. So that's terrific. We can use sequencing now for a lot of things where we used stuff like genotyping or microarrays, and sequencing gives us much better results. But it also poses a problem because if you do an experiment with microarrays, it might generate like megabytes of data, but if you do the same...
Oh, great.
OK. Yes. So yeah. Whereas 10 years ago, if you did an experiment with a microarray, I was saying it generates megabytes of data. Whereas now, doing the same experiment, the same gene expression survey, for example, using sequencing, it will generate hundreds of gigabytes of data. So this is a challenge because whereas using pure Perl or Python is a perfectly practical way of analyzing data in that generation, now it becomes impractical very quickly. Yeah, so this talk is going to be something of a preview of the core package that the BioJulia organization is working on called BioJL. So some of what I'm about to show is stuff that's still sitting in pull requests or different branches. We've generally been pretty conservative about merging stuff into master, trying to make sure everything's tested and people have a chance to do code reviews. But all of this works now. So one of the first problems we started to tackle was the problem of parsing. Bioinformatics, I'm sure it's this way in a lot of other fields, it contains a lot of ad hoc file formats that arise basically because someone wrote a program, they came up with some file format for that program to write to, that program became popular, and now everyone else is using that format. And it becomes a problem because a lot of these formats there's no specification for. So when people do produce specifications, it's more observing, here's what the different programs will parse rather than a prescriptive definition of this is what this format is. So here's a particular case study. This is one of the most ubiquitous formats called FASTA, or FASTA, some people say. And it's simply a text format to store sequences. It just looks like this. There's a line here to indicate some identifier or name of the sequence, and then there's just a bunch of plain text DNA sequence here. Right, so it's a pretty simple format, and there's tons of tools that can process it. Like here's a command line tool which will take as input this sequence and return the reverse complement. So DNA, it's double-stranded, so one strand has a complementary strand going in the opposite direction. So you can compute that transformation. A lot of tools do that. So the problem is that people tend to write parsers, because the format is so vaguely defined, that's kind of an excuse to write a parser that's equally vague in what it accepts. So for example, this tool, which it's a pretty popular tool, it won't return a parser error on anything. You can feed it any input, and it'll be fine with that. And to prove that, so this is an ASCII art pizza. I was trying to find the furthest text file from the fastest sequence file I could find. And sure enough, if you asked for the reverse complement of this pizza, it throws the first half of the pizza away, and then it splits the second half of the pizza into what it thinks is two sequences, and then just gives you a bunch of garbage. So I mean, it's obvious that the pizza is bad input, but there's no specification for FASTA. So if there's something subtly wrong with it, this tool is just going to skip over a bunch of your data and not tell you. But this is like far from a straw man or like worst case scenario, because this is actually, the parser that's used in this is one of the most popular parsers that people use in C and C++ projects for FASTA. So I searched on GitHub and it's used 1,600 times. Yeah, so when we set out to write parsers for our package, so stuff like this, it's not hard to write a parser by hand to read this. But we'd like to avoid that, partly because these are so ill-defined. We really want to work at the level of the definition of the format rather than at the level of writing code to try to parse it. Like when it's vaguely defined and you're setting out to write code to parse it, your parser often ends up kind of vague. And often only one or two people understand how the parser works, so you can't really argue about what are the limits of this format. So yeah, so the approach we've taken is to try to generate all of our parsers from specifications. We're using a tool called Raggle to do that, which is it takes regular expressions and generates code and whatever host language to parse those. So obviously Julia has regular expression support built in and with PCRE, but Raggle gives us some nice features that we might not as easily get. So the first issue why we didn't use PCRE is because even with name submatches and things like that, once you start writing a very complicated regex for PCRE, it gets hard to understand pretty quickly. So we kind of wanted something with a nicer syntax. And then there's also some practical concerns, the biggest of which is a lot of these files we want to avoid reading the entire thing into memory before matching it. And at the same time, it's not necessarily line-oriented. So we want to be able to run the state machine until we hit the end of the input and then read more input into the buffer and then continue running from where we left off, which is not something that's so easy to do with PCRE, although I think it might be possible. Yeah, so to write a FASTA parser in Raggle, it actually looks very nice. This is copy and pasted from our code. Let's zoom out a little bit. Yeah, so I mean, it's very nice because we name things. You can kind of read this from bottom to top. So main is basically a pattern for the entire file that we're trying to match. And the file is just any amount of leading white space plus any amount of FASTA entries. FASTA entries consist of this character with some identifier and maybe a description and then a bunch of sequence. Yeah, so it's quite readable. You just have to know a little bit about regular expressions, and you can modify these parsers if you disagree on what we define as FASTA. Yeah, so Raggle's very nice in that it's designed to support multiple host languages. The current version, how it works, it's pretty neat. So it'll actually generate a parser for this language in kind of a C-like pseudocode, and then they have a separate step that processes the AST for that pseudocode into the host languages. So to support Julia, all we had to do is write a backend that transforms that code into Julia code, which is quite easy. Oh, and then the other part was to get go-to statements into the language, which was a little work too. Yeah, so one of the really cool things that Raggle gives us is that we can embed arbitrary Julia code into the state machine. So like here's a trivial example. We want to count line numbers so we can say if there's a parser error, what line it occurred on. So we can define an action here in Raggle, which is just some arbitrary Julia code. And then we annotate this new line portion of the state machine with this, which says, when you enter a state that's associated with new line, execute this code. And then we can just keep track of line numbers that way. But this is also very nice for extracting portions of the input from what we're parsing. So we don't want to have to do any unnecessary copying. So we match portions basically like this. So this annotation means when you enter one of these states, execute this code. And what we do is mark the position in the buffer at what point we enter that state. And this says when you leave that state, execute this other code. So we execute this code, which says, OK, now take this span from the buffer. So So there's these weird macros because we do some tricks where if we refill the buffer, we want to update that position and keep it in the buffer and that sort of thing. But yeah, but this is generally quite efficient in that we don't have to, build temporary buffers to, keep track of things for the most part. We can just extract it straight from the read buffer. This also comes in handy because we can parse. So, ostensibly it's a regular expression parser, but we can do some tricks to parse things that are a little bit more than regular. So the vast majority of the sequencing data that's being generated is in a format called FASTQ, which, zoom out so you can see this better. Yeah, so FASTQ looks like that, and it's it's supposed to be analogous to FASTA. It has some read identifier. The sequencer only reads little fragments, so this is a DNA fragment that the sequencer read. And then this line here is quality scores, which is essentially the sequencer assigning a confidence level to how sure it is that it called these positions correctly. OK, so that's fine. Like this format, its only job is to store 3 strings per entry. But it's so poorly designed that you actually can't parse this with a regular expression. So to see why, like you see, so the identifiers start with this @ symbol, yet, so these are ASCII encoded quality scores. It's maybe hard to see, but the @ symbol is actually a valid quality score. So we don't know when to stop matching quality scores until we've reached the same length as the sequence. So it's actually not a regular language if you're parsing it correctly. But of course, there's no specification for FASTQ, and actually not very many people realize that it's not a regular language. So there's plenty of incorrect parsers for FASTQ. But there's a cool trick that Ragel can do, which is this when statement here, which is only take a state transition if a particular condition is true. And the condition is written in Julia. So here we're just saying, once we've consumed the same number of quality scores as sequence scores we previously consumed, then you're allowed to take this transition. So it's like a context-free language, but we're parsing it with like a hacked regular expression parser. Oh, I bumped it again. Yeah, so the one downside is Ragel generates Julia code, but it's pretty inscrutable Julia code. It's like, you'll get like 500 or 1,000 lines that just look like this and go on and on and on. So it's inscrutable because, it's a bunch of spaghetti code where it's, following a bunch of go-to statements. But at the same time, it's actually not very sophisticated code in that, like, most of the code is just, compare some byte in the input, and then, if some condition is true, follow this go-to, otherwise follow this go-to, and so on. So, I mean, it's hard to read this and, like, debug anything or profile anything, but fortunately, we can get state machine diagrams out of this. So, as I was doing parsers, I was finding, a much better way to try to optimize these is just generate the state machine diagram and see if there's any unnecessary states that we can remove. So this is quite nice. So yeah, so this is the diagram for our FASTA parser. Yeah, so if you're not familiar with these state machine diagrams, it's just going to accept the input one character at a time, and based on what that character is, follow one of these lines into a different state. Yeah, and then it accepts if it's in one of these states that has 2 circles. And the good news is that they're pretty fast for the most part. So here's us reading in the human genome in FASTA format, and it took us about 26 seconds, which is a little bit faster than BioStrings, which is a popular R package, or seqan, which is a C++ project, or C++ package. So I'm going to show a bunch of benchmarks against BioString and seqan. There's a lot of sequence implementations I could test this against, but these are among the fastest. Oh, so BioStrings, it's an R package, but this is kind of a fair comparison because, like most high-performance R packages, it's mostly C code with an R interface. Oh, they're also quite versatile. We have some nice features. You can read stuff like a regular file, but you can also you can memory map things. You can read things from commands. Here I've compressed some file with my own compression algorithm, but I don't have to write Julia bindings because I can just read it from a command. Oh, I wanted to show this off. One thing we do, so like FastQ has these ASCII encoded quality strings, which are unreadable. So we instead print it as these nice Unicode bar charts. Yeah, so it's also a challenge to verify the correctness of these parsers, since not everyone has the same idea of what the format is. So the approach we've taken is, you can either think of it as being very rigorous with testing or being very lazy with testing. When we were looking at other packages for what they test against, we realized that it's like completely disjoint sets of test cases. So we made our own package called bioformat specimens, and we just started ransacking test cases from other projects. So our parsers are, kind of by definition tested on a proper superset of all the test cases in BioRuby, BioPython, and BioPerl, because we just took all their test cases and indexed them. Right, so we have a few parsers written, and we're starting to write more. So we'll move on to how we're representing nucleotide sequences. So when we're surveying packages, the most common way to represent a nucleotide sequence is, just basically it's just a tuple, a string, and then some alphabet associated with that string. This incurred a lot of overhead because it's kind of like having an alphabet associated. It's like having a type, but it's like a type you have to constantly be checking. Like anytime you do something, you have to check, okay, am I in an alphabet that I can use to do this? And plus, if you want to be very rigorous, you want to see if there's any characters in that string that aren't in the alphabet, and so on. So it's quite an overhead just in that regard. I mean, in terms of performance, and also just in terms of remembering to check the right things when you're writing code. So what we did is slightly unusual. There's 4 nucleotides, so you can obviously 2-bit encode these, and we're, of course, not the first person to think of 2-bit encoding nucleotide sequences. But somewhat unusual to do this as, the one representation of nucleotide sequences in a general purpose package. Right, so 2-bit encoding, there's an obvious encoding for nucleotides. But there's also the practical concern that there's 4 nucleotides, but in practice you also have ends representing NA, because often there's things that can't be sequenced, like very repetitive DNA is hard to sequence, so there'll be stretches of ends in the human genome sequence. So what we do is we have our 2-bit encoding of the sequence, and then we have a separate bit mask basically for ends. So in effect it's 3-bit encoding, but we found it useful to split them up rather than try to pack 3 bits in a string. Often you can implement algorithms very efficiently by assuming that there are no ends and running that algorithm on the 2-bit sequence, and then going back and fixing it up by processing the ends. So we incur kind of a encoding tax whenever we read in sequences, because actually this 2-bit encoding takes a little bit of time. But in exchange, we think it's a worthwhile trade-off because there's a lot of very basic sequence manipulation that becomes really fast. So like here's computing the reverse complement, and we're much faster than anyone else basically, because you can do some cool bit twiddling tricks to do that. Oh, actually, the bit twiddling tricks are pretty obvious. So it's reverse complement, which is reversing the bit string and taking the complement, and the encoding is such the way that the complement in the DNA sequence terms is the bitwise complement. So that's very easy to compute. So like here's counting short oligonucleotides or k-mers. So we want to just build a table of all how many times each, in this case, 10-mer occurs. And we can do that quite a bit faster than either biostrings or CCAN. Then counting nucleotides, we can do that way, way faster than anyone else. So this taking advantage of our 2-bit encoding. So like, to get a sense of what we're doing, so like this is how you count Cs in a nucleotide sequence encoded in an integer. So it's just a handful of instructions to count how many Cs are in a 32 nucleotide sequence. Right, so that's the native code. So you can imagine like counting Cs over a long sequence like the human genome. It's a very small number of instructions compared to the naive method, which would be comparing each character and saying is this a C? If so, increment this counter and so on. Here's translation. So I took all the protein coding DNA from the human genome and translated into amino acid sequences. So we can also exploit the encoding to do this a fair amount faster than anyone else. So the other important thing I think about the design of our sequences is that we've done it in such a way that sequences and subsequences are the same thing. So subsequences are cheap. They share the same underlying data. And we can do this because they're immutable. So you can do sort of the naive thing of processing a DNA string by taking a whole bunch of different subsequences and not worry too much about time or memory. So like this is the first million suffixes of chromosome 1, which would, naively, if you're to do this by copying, it would allocate, I think, over a terabyte. But here we can do it in milliseconds and not really allocate that much at all. So this is, my notebook's a little bit out of date. But yeah, so when we take subsequences, it's not like this second-class type that's a subsequences. A sequence and subsequence are the same thing. If you take a subsequence, you get a sequence. So it's not like a second-class citizen. You can run any function on a subsequence that you can on a sequence. Yeah, so the goal of a lot of this work is that there's sort of this artificial division that you see in a lot of bioinformatics software, where there's kind of like stuff that's written in applications that can be written in R and Python and Perl and stuff like that. And then there's an entirely separate, non-overlapping set of problems that are almost always implemented in C or C++. So a lot of the impetus for doing this in Julia is we kind of want one language that can span that spectrum, where you can do sort of interactive analysis work, but also write heavy-duty kind of stuff that's pretty much always done in C++ now. So just as a proof of concept, you can write a DNA assembler with what we already have. So let me explain the problem. Yeah, so I showed you a little bit of those sequence fragments that come off of sequencers. So basically the problem with sequencing technology right now is what we want to do is take a long sequence and just treat it like it's a magnetic tape and just read the nucleotides off of that. But the current technology that's used, or the most popular technology, has the property that as you go along reading it, the error rate kind of shoots up exponentially. So really you can, in practical terms, you can only accurately read short, like less than 100 nucleotide fragments at a time. So the way we sequence genomes is essentially, in effect by, if it was a magnetic tape, is randomly moving the head around and restarting the reading process. And you do this by just making a bunch of copies of the sequence, of the sequence, and then randomly fragmenting it, and then sequencing all those short random fragments. So then you have a bunch of short sequences that you've sequenced pretty accurately, and you have to try to reassemble what you started with. You can ignore that one. So one cool approach that is pretty popular to do this is based on the notion of de Broglie graphs. So de Broglie graphs, so in its original formulation, it's often showed in binary alphabet or bit strings. And what it is so if it's in binary, you take every binary string of some size as the nodes in the graph, and then there's an edge. There's an edge in the graph if you can get from one node to the other by shifting it by one place and adding either a 0 or 1. So like, let's see, where should I start? So like 0, 0, 1, you can get to 0, 0, 0 by shifting it right and adding a 0 at the end. So this is like a complete de Broglie graph. It has every sequence, every string over the alphabet of some fixed size. So how this relates to DNA sequencing. So we have a bunch of short fragments. So of course this can be over any alphabet, not necessarily binary. So if we do it over ACTG, DNA sequence, you can build a graph like this, and if you sequence a genome, some subgraph of the full de Broglie graph will be evoked, right? Because we just observe some of the DNA sequences. And then if you assume there's no errors in the sequencing process, which is not a good assumption, but let's assume it anyway, you can recover the full sequence that you started with by finding an Eulerian circuit, an Eulerian path through the de Broglie graph, which is a path that visits every edge exactly once. So there's often more than one such path, but the string you started out with will be one of those paths, and you can try to use a heuristic to sort out which. OK, so here's a de Broglie graph assembler in well, less than 40 lines of Julia. It's a toy assembler. You couldn't use it for anything practical, but it's at least a proof of concept that with more specialized data structures, you could write this kind of stuff in Julia. So all it involves is I'm counting k-mers and a bunch of sequencing reads. And then I'm trying to build a long contiguous paths in this by just starting with some seed k-mer, what I'm doing here, and just trying to greedily add the next most abundant k-mer and keep following that greedy path and try to build a long sequence. So it's pretty simple, and I'm using the base Julia dict, which is good, but if you're doing this real, you probably have a more specialized hash table. And the greedy approach is a pretty poor heuristic for this. But so here's an experiment. Here's 10,000 reads from a data set I had lying around, and we can, it's not great, but we can generate a 161 nucleotide contig from that. Yeah, so I mean, it's obviously a toy example, but because this is like a laughably small number of reads to do assembly with. And also this data set is, it's from acid mine runoff, which you can imagine there's not very many species that live in acid mine runoff, so it's a very non-diverse set, so there's less sequence to assemble. But yeah, as a demonstration, I think we have all the parts that you need to write an assembler. So I hope stuff like assembly and alignment can be done in Julia, because a lot of the same stuff gets implemented in C++ over and over, and it's much easier to share code this way. The second big project we've been working on is representing interval data. The nice thing about genomics is that a great deal of the data is essentially one-dimensional. You're measuring something across a very long string. So it's nice that it's one-dimensional, but there's also a great deal of it, and you want to work with it at different scales. So interval data is a lot of what we work with in practice. So basically, to be concrete, what I mean about intervals is it's some sequence and some closed interval on that sequence. Sometimes there's a strand associated with it, one strand of the DNA or other. And then you associate some data with that interval. And we need to be able to process large amounts of that. And kind of the most fundamental thing we might want to do with interval data is intersection, which occurs all the time, which is you have two data sets consisting of intervals, and you want to return all pairs of intervals, either an A and B that intersect. Yeah, so this happens a lot. Like you run two experiments and you get a different set of intervals, and you want to say, oh, how do these compare? Or like you sequence reads and you want to see what genes they overlap, or that sort of thing. Right, so the interesting thing about doing intersection is there's actually multiple ways we can go about this. If we index one of these or the other, we're able to do log n single interval queries. So in other words, if I have an interval and I want to see how many intervals in A it intersects, I can do that in log n time. So I could just process all of these intervals against A and do, well, the size of B times in each query would be an order log the size of A. And of course, intersection is symmetric, so we can flip it and do it the other way and get essentially A log B time. But the other way we can do this is process this in sorted order. If we assume the two lists of intervals are sorted, we can do something like, sort of, if you're familiar with the merge operation for merge sort, is just start from either end and walk our way through these two sorted sets of intervals looking for intersections, and do that essentially in linear time. But which approach you take depends very much on the size of the two sets of intervals. Most of the time, doing the linear search is the fastest way, but if you have one very big set of intervals and the other one's very small, it makes much more sense to do this log n query thing. So we're trying to come up with a data structure that's does everything well, essentially. And what we settled on was something based on B plus trees. So B plus trees have the very nice property that, so this is a very schematic representation of B plus trees. But it's a balanced binary search tree in which you have nodes that store many keys. And in a B plus tree, the internal nodes store only keys, whereas all the data is stored in the leaf nodes. So it's very nice. So we want to be able to do these log-n queries efficiently, but we also want to process it in sorted order as efficiently possible, as efficiently as possible. So with B plus trees, you have these large continuous block, these leaf nodes that have a bunch of data in sorted order. So we can basically iterate through these. So this is schematic. In practice, we're using 64 intervals per leaf node. So you can iterate through it. You only have to follow one of these pointers, every 65 or so items that you're iterating through. So it's not quite enough just to have a search tree. It has to be kind of an augmented search tree, because you can imagine if you're doing interval queries, you also have to know the end node. Otherwise, it's not log n because well, you can't remove anything from the search from the beginning, because you could have an interval at the very beginning that spans the entire sequence. So you always have to search at the beginning for the first interval, and it ends up being linear time, unless you augment this tree with the maximum endpoint in each subtree. So it's sort of a little bit tricky binary search tree, but it's essentially a binary search tree. Yeah, so we can do intersection queries very efficiently. So I'm benchmarking against this very popular tool called bed tools, which is a mature C++ project. It's a command line tool where you input, the format here is called bed. I actually don't know the reason why it's called bed. But yeah, so you input bed files, and it can do a lot of different operations like intersection and merge and stuff. And it's generally thought to be quite efficient. But with our interval collection implementation, we can actually beat it at a lot of things. So we're doing this a little bit faster. So I have some sequencing reads, and I also have some gene annotations. The gene annotations are here's the position in the genome where genes that we know about are. So this creates an iterator over these genes. Here I'm indexing all the reads, and I'm performing a bunch of queries against those reads, which is essentially the same thing that bed tools is doing here. Of course, we can process these in sorted order sometimes, so we don't want to have to index it if the files are already sorted, which is often the case. So we also introduced the notion of an interval stream, which is just an iterator over intervals. An iterator over intervals where we're assuming that it's in sorted order. So you can see we can do the same sort of intersection here very efficiently and never read the entire data set into memory. So these read operations return iterators over the entries in this file. So we're doing this intersection in about 80 seconds, where bed tools takes about 147 seconds. Maybe I'll skip ahead a little bit. Just one last thing I wanted to talk about. So genomics, it involves visualizing a large amount of essentially one-dimensional data. And there's a common idiom that evolved to do that, which is the genome browser. And often they look something like this. And this is displaying some segment of the genome, and it's, you can put a lot of different data on it. In this case, these are some gene annotations, and this is the human genome, and it's aligned against a bunch of different species, and it's showing how similar they are. So like we're very similar to chimp and bonobo, and somewhat less similar to mouse, although there's a bunch of blocks that match, and so on. So yeah, so this is nice. You can zoom in and out, and it's, it does that. So yeah, it's not super responsive because of it's reading stuff from different servers, and it's rendering PNG files on the server and then serving them up. So we really like this idiom, but we also like Jupyter notebooks. So we figured we can, well, this demo might not work in time. Maybe I have another one open. OK, well, I'm not going to be able to do this demo in time, but we basically figured out how to put genome browser and the Jupyter notebook using this project called Dalliance. So yeah, so it looks basically like this, but you can imagine in a Jupyter notebook. But the cool thing is that we can use data directly without having to write it. So this works, it's a client-side genome browser, but it works by reading data from a server. So to visualize anything on this, you have to write your data to the file in the right format and put it up on some public server. But we're doing it directly in memory in the genome browser, or in the Jupyter notebook, which is pretty cool. You can see this is very responsive. And since we're doing it all in memory using some fancy file formats, it's actually much more responsive with this, which I wanted to demo, but notebook glitches. Yeah, so I guess I'll end there, and I'm happy to answer questions. Oh, wait, let me say one more thing. There's I should thank, this is not just my project. There's a bunch of people that have worked on this. Ben, Richard, Paulo in particular, and also Kenta, who's here, and he's joining us for Julia Summer of Code and working on some cool alignment stuff. So thanks to them for supporting this as well. OK, so I'll end there. Thanks. Yeah.
I noticed you're spending a lot of time in garbage collect. Is that in version 3 or version 0.4 of Julia?
This is version 4. Yeah. So our parsers now are like returning a new entry for everything it sees in the file. So something like reading reads, it does end up allocating a lot. That's a very convenient thing to do. So we're thinking about like maybe also implementing readers that are mutable, so it doesn't allocate and just overwrite something to do as fast as possible. Although we've been pretty happy with our convenient yet a little bit wasteful parsers at this point. We're also not done tuning the GC, yeah. So, yeah.
Why you went to 2-bit stacked instead of like 2 separate arrays, so you could mask directly between the 3?
Oh,
where you'd have the same bit masked for walking over.
Oh, I see.
One array for one. In my bit twiddling experience, that might be faster for some of your operations.
That's something to consider. I don't know for sure whether that'd be faster or not. I mean, you get some advantages from caching, but
Offline we can talk about it.
Yeah, sure.
Okay. Epigenetic markers, are you doing anything with that?
I'm not personally, but I'm sure there are people who are interested. Yeah. Okay, thanks.